A primary goal in designing experiments is making it easy to measure real effects while minimizing the effects of confounding variables. One basic way of doing that is with a randomized experiment. As described in the previous section, a randomized experiment is done by selecting random samples from the population, applying different treatments to each sample, and comparing the effects of those treatments. The effects of confounding variables are minimized in a randomized experiment because the samples, at least on average, are all affected equally by confounding variables, so that the only systematic difference between the samples is the treatment. It is important in an experiment that all of the samples be treated equally during the experiment. For example, in an experiment to measure the effect of treating plants with nutrients, if the treated plants receive different care, or if the method used to apply the nutrients has other effects, such as adding water or disturbing the soil, or if the explanatory variable is measured differently for treated plants, those differences are confounding variables that could affect the results. To reduce the effects of differences that accompany the treatment, it is common to apply an inert treatment called a placebo to the non-treatment sample, such as by applying an inert treatment to the control group using the same process as is used for applying real nutrients to the treatment group. To reduce the effects of differences such as differences in plant care or differences in how results are measured, it is common to arrange for individuals in the treatment group to be indistinguishable from individuals in the control group so that any differences in care or differences in measurements are spread equally between the groups. A double-blind experiment is an experiment that includes both of these changes, using placebo treatments so that all groups are treated as similarly as possible, and concealing any distinction between the treatment group and the control group, such as by making treated and untreated plants indistinguishable to anyone who cares for the plants or who collects data. Randomization is usually the only way of distributing unknown confounding variables evenly between samples, but when confounding variables involve known differences, it is possible to improve on randomization by organizing the population into groups of similar individuals and doing separate randomized experiments within each group. Groups of similar individuals in an experiment are called blocks, and the variable that is shared by the individuals in a block is called the blocking variable. For example, in the plant nutrient experiment, the type of plant could be a blocking variable. Organizing similar plants into blocks and doing a separate randomized experiment in each block has the desired effect of arranging experiments in which the treatment group and the control group in each experiment have similar distributions of the blocking variable. Blocking can also be done for more than one blocking variable, such as, for example, by organizing plants into blocks based on the type of plant and on whether the plant is placed in shade or in direct sunlight. The advantage of blocking over randomization is similar to the advantage of stratification over simple random sampling. While randomization can only make groups similar on average, blocking can make groups nearly identical, at least with respect to the blocking variable, not just similar on average. A matched pair experiment is a special case of a blocked design in which each block has only two individuals. A matched pair experiment could be set up, for example, by arranging similar plants in pairs and randomly treating one member of each pair with nutrients and the other member with a placebo. With a matched pair design, it is easier to consider many different blocking variables since it is only necessary to find two individuals with similar characteristics.